Hello, my name is Howard Montoya, and my topic will be on natural killer cells and HIV pathogens. What are they? Natural killer cells, also known as NK cells, they are lymphocytes in the same family as both T and B cells. They are the body's mechanism of defense against foreign invaders. They are best known for killing virally infected cells and detecting and controlling early signs of cancer. They were named natural killer cells because they do not require activation to kill cells that are missing self-markers of MHC class I. They are the most aggressive of all lymphocytes and have no immunological memory. They are part of your innate immune system, also known as specific or non-specific immunity. What types of cells are they? They are not phagocytic and are one of three kinds of cells differentiated from the common lymphoid progenitor. NK cells belong to a group of innate lymphoid cells. They are known to differentiate and mature in the bone marrow, lymph nodes, spleen, tonsils, and thymus. Their granules contain preformed biologically potent molecules and play a role in defense against both intracellular and extracellular pathogens. What do they do? The role of NK cells is analogous to the cytotoxic T cells in the vertebrate adaptive immune response. NK cells provide rapid responses to viruses and infected cells, acting at around 3 days after infection and respond to tumor formation. NK cells are different from typical immune cells because they have the ability to recognize the kill stress cells in the absence of antibodies and MHC1. In contrast to natural killer T cells, NK cells do not express T cell antigen receptors, pan-T markers CD3, or surface immunoglobulin B cell receptors. They patrol your body and lymph looking for abnormal cells and are unique in the aspect that they can kill a person's own cells if they have become infected with a virus or become cancerous. They kill on contact by binding to its target. When a cell becomes infected, they stop making major histocompatibility complex on their surface. Once a cell is infected, it secretes type 1 interferons, which binds to the receptors of the NK cells. As a result, they become activated, proliferated, and differentiated into effector NK cells. When they detect this, they do not perform macrophage, the act of swallowing and breaking down infected cells or virus. Instead, they bind to the infected cell and poke it with an enzyme that triggers holes in the cell's membrane known as apoptosis, programmed cell death. After a natural killer cell has destroyed an organism or virus, it displays the pieces on the pathogen on its outer layer to help better combat similar pathogens later in the future if it encounters it. They make approximately about 5 to 15% of the lymphocyte population. What is a pathogen? A pathogen is a microorganism that can produce an infection or disease. It can be anything from a virus, bacteria, fungi, or even protists. The term is used to describe an infectious microorganism or agent. The scientific study of microscopic organisms, including microscopic pathogenic organisms, is called microbiology, while the study of disease that may include these pathogens is called pathology. HIV. What is HIV? Human immunodeficiency virus, also known as HIV, comes in two species or strains of the virus, type 1 and type 2, that both infect humans. Although type 2 is more seldom, over time, they weaken the body's immune defense by attacking the immune system, ultimately leading to acquired immuno disorder or immunodeficiency disorder, also known as AIDS. In most cases, HIV is a sexually transmitted infection, STI, and occurs by contact with body fluids, blood, or shared needles. Non-sexual transmission can occur from an infected mother to her infant during pregnancy, nursing, or childbirth. HIV infects vital cells in the human immune system by attacking helper T cells, macrophages, and dendritic cells that are all part of the humoral response of the immune system. The outer envelope of HIV is covered in glycoproteins, which mutates frequently, thus making it hard for helper T cells to detect them. In order for HIV to gain access to a cell, it has to attach to both the CD4 and the co-receptor. Once attached to specific receptors on the T cells, it begins to fuse the membranes together, where it then releases 2 RNA strands and 3 essential replication enzymes. It is then transcribed into DNA and integrated into the host genome, causing the T cell to treat it as its own and making more copies of the virus. In this process, it blocks CD4 glycoproteins on the T cells, which stops it from communicating with other response immune cells known as antigen presenting cells, APC, such as your dendritic and macrophage cells. After copies have been made from the host cell, it then leaves the cells and matures, infecting more and more T cells, macrophages, and dendritic cells. When the body enters the chronic phase, it starts to lose roughly around 1 to 2 billion T cells a day. NK cells and HIV research. NK cells play a crucial role in viral immunity. Specifically, NK cells directly mediate immune pressure leading to virus evolution. NK cells receptors, genotypic profiles, clonal repertoires, and functional capacity have all been implicated in virus containment. Natural killer cells function then becomes abnormal, thus allowing HIV to multiply inside human cells. When the virus replicates itself, it does not make an exact copy, but an off copy, allowing it to target different parts within the human body. HIV is hard to treat because it mutates at a high rate. More than 10 billion virions are created each day. During these initial stages, known as the latency period, a person may not show symptoms for up to 2 to 10 years. If not treated, HIV kills off the T cell it infects. When the T cell count falls below 200 cubic meters of blood, it then becomes acquired immunodeficiency syndrome, also known as AIDS. After the immune system becomes that oppressed, a person does not die from AIDS, but instead opportunistic infections that the body can no longer fight off, such as cancers, encephalitis, tuberculosis, or even pneumonia. An opportunistic infection is when infection starts to occur more frequently due to a weakened immune system that can become severe and lead to death. Those who do not have HIV and are at high risk of contracting the virus can take prophylactic, also known as PrEP, which works similar to an antiretroviral drug by blocking the enzyme reverse transcriptase, in which the virus then has trouble binding to its host. In conclusion, we should all take microbiology and pathology serious, especially in light of recent events concerning the COVID-19. HIV and AIDS research continues to be a necessary endeavor until a cure is found.